Hello, everyone. My name is Ayesha Ali Khan. I'm a student at the Department of Chemistry and Biochemistry, and today I'll be presenting my work on how oncogenic MYC hyperactivation affects nerve biogenesis and exposes vulnerabilities in the spliceosome with implications in cancer biology. So to begin, MYC is a proto-oncogene that regulates essential cellular processes like transcription, metabolism, and cell proliferation. Under normal conditions, MYC maintains a balanced transcription levels, but however, in cancers, MYC is hyperactivated, leading to transcription amplification. This dramatically increases pre-mRNA levels and puts an increased demand on the spliceosome, which is a complex responsible for removing introns from pre-mRNA, and it is an essential step in the maturation of the pre-mRNA. The spliceosome is made up of small nuclear RNAs called snRNAs and its associated proteins, which is collectively known as the SNRPs, small nuclear ribonucleoproteins. Oncogenic MYC increases pre-mRNA transcription, placing a higher burden on the spliceosome, which becomes essential for cell survival. Depletion or inhibition of the core spliceosome components, such as BUT31, disrupt splicing, leading to apoptosis in these cells. So this dependency on an intact spliceosome makes MYC-driven cancers highly vulnerable, presenting a potential therapeutic target by exploiting spliceosome inhibition. And we believe that this imbalance in the SNRP repertoire, which is the level of the different SNRPs, may lead to aberrant mRNA splicing. And our study is focused on the SMN complex, which assembles the SM ring onto the snRNAs, which is a critical step in the SNRP maturation. So to investigate this, we used MYC ER-HME cells, which allow MYC hyperactivation upon 4-hydroxytamoxifen. 4-hydroxytamoxifen or 4-OHT is an active metabolite of tamoxifen, which is a selective estrogen receptor modulator. It binds to estrogen receptors and can have an agonistic or antagonistic effect depending on the cellular context. For example, in breast cancer cells, or as in the model that we're using, where estrogen receptors are fused with other proteins such as the MYC-ER fusion systems, 4-OHT can function as an agonist, leading to the activation of downstream target genes like MYC. So, our data shows that MYC hyperactivation leads to an increased expression of several SNRP-associated proteins like the SMN, Gemin-5, SMBB prime, as shown in the Western blot in the top right. However, the increase is not uniform, which may contribute to the imbalance in SNRP levels. Further, using RT-qPCR, we observed that SNRP repertoire shifts under MYC hyperactivation. This supports our hypothesis that MYC alters the splicing environment, potentially leading to aberrant splicing of critical genes. So looking ahead, our future directions include assessing SM core assembly more directly using immunoprecipitation and Western blotting. We also plan to examine the role of mTOR signaling, particularly its involvement in regulating SNRP biogenesis under MYC hyperactivation. So in summary, our work suggests that MYC hyperactivation stresses the spliceosome machinery, especially the SNRP biogenesis pathway, revealing a potential vulnerability in MYC-driven cancers. So this could lead to novel therapeutic strategies that target splicing machinery to combat oncogenic MYC activity. Thank you for listening to my presentation. I'd like to thank my collaborators, Dr. Young and Dr. Westbrook and Dr. Bowling. And I'd also like to thank the chemistry department of UTA, the National Institute of Health, and my PI, Dr. So, for giving me this opportunity to do this amazing research. Thank you so much, and I'll be looking forward to any questions that you all might have for me.